what we have to do this morning is try and understand how different types of viruses replicate their genome and express their viral genes inside the infected cell. And this is really very closely related to the Baltimore class of different virus types, because depending on whether your genome is DNA or RNA plus strand or minus strand, then you have to use a different strategy to replicate and express the genome. So I'll give a little introduction about that, and then we'll look at a couple of examples from DNA viruses, a couple of examples from RNA viruses, and then maybe end up with retroviruses, which are kind of a strange mix of both of them. So what we're doing is really thinking about this part of the replication cycle, the gene expression and genome replication. So thinking about Baltimore class, so this was a slide I prepared really a long time ago now. And the idea is to try and, so the Roman numerals, this is the Baltimore class, and the size of the typeface is correlated with the number of different virus families within each Baltimore class. So the most, and then the little symbols nearby are the types of capsid structure you get within each Baltimore class. So you can see that the two most diverse types of viruses are those with double-stranded DNA genomes and those with plus-strand RNA genomes. And that's really because what these guys are trying to do here is, okay, they were getting inside an infected cell, and they try and convince the infected cell that the virus genome really is just like cellular genes, and the cell can express those genes. Whereas what the plus-strand RNA viruses are doing is they're generally passing themselves off as a messenger RNA. And they have to get the host ribosomes to express proteins from their genomic RNA. And these are the two main strategies. So basically, most DNA viruses and the retrotranscribing viruses replicate in the nucleus, because their basic idea is we're going to act like a bit of the cell's DNA. The cell's DNA is in the nucleus, whereas the RNA viruses, they generally replicate in the cytoplasm. Now, there are exceptions in both directions. So there are some DNA viruses that replicate in the cytoplasm, and there are some RNA viruses that replicate in the nucleus. But in most cases, DNA viruses in the nucleus, RNA viruses in the cytoplasm. So we're going to think about DNA viruses first. So what do they have to do? So they've got their genome is in double-stranded DNA, and to replicate the genome, they've got to have DNA replication. So that requires specialized enzymes, DNA polymerase, but not also that, also a primase enzyme, a helicase, and other accessory proteins. And it also requires the substrate for the DNA polymerase, deoxynucleotide triphosphates, which may be present in the cell at high concentrations or might not be. Now, host cells can perhaps produce many of these things, but when virus DNA replicates, all of the DNA replication machinery has got to be directed towards virus replication. So it's got to be directed towards the viral origin of replication. For genome expression, that's kind of easy for DNA viruses, especially if they replicate in the nucleus, because DNA viruses that replicate in the nucleus, they can use host cell transcription factors to initiate transcription of their genes from the viral promoter. They can use RNA polymerase II from the host. They can use the enzymes required for mRNA maturation and splicing. So if the DNA virus replicates in the nucleus, all of these molecular mechanisms are available in the host cell. Okay, so DNA replication required, mRNA expression required. Now, this is going to be the first choice for, well, choice, you know, if you see what I mean, for a DNA virus. So if you were trying to design a DNA virus from scratch, this would be your kind of like decision-making process. Is my virus going to replicate in the nucleus? So if the answer is no, then into that virus genome, you would have to put all the genes coding for enzymes required for DNA replication and for gene expression. So you'd have to have a DNA polymerase, you'd have to have a primase, helicase, accessory proteins. You would also have to have an RNA polymerase and your own transcription factors. And of course, this would take up a lot of space in the genome. So it's only the very largest DNA viruses that can do this. So these are the pox viruses, and then the giant DNA viruses like mimivirus or Pandora virus. Okay, so these are what they call large nucleocytoplasmic DNA viruses. They can replicate in the cytoplasm, and they are not dependent on the host cell for either DNA replication or genome expression. Okay, so what about, okay, sounds a bit complicated. How about if I replicate in the nucleus? So then it kind of depends what cell you are infecting. If you infect a rapidly dividing cell, then the host cell will already be in the cell cycle, and it will already be expressing all the proteins required for DNA replication as well as RNA transcription. So the virus does not have to do anything really. And so there are some small single-stranded DNA viruses that can infect us. Parvovirus B19 causes a childhood illness, and it infects, I think it's erythroblasts, but the hematopoietic precursor cells that produce erythrocytes. And these cells are dividing continually every day. And so once the virus infects these cells, well, you know, DNA replication machinery is there, RNA transcription machinery is there. It works yes.
Does a bigger virus will struggle more to enter the cells than a smaller?
Can a bigger virus get into the nucleus? Is there some upper limit? So for example, the herpes viruses, they've got genomes might be 200 kilobases. And if you remember, they would dock at the nuclear entry port and inject the DNA. So it's not like the whole particle has to get into the nucleus, only the DNA has got to get into the nucleus. So I would say, is there an upper limit on this process? Probably not a very strict one. Okay. So if you can infect a rapidly dividing cell, it makes your job easier. But of course, most of the cells in our body are not rapidly dividing. Okay, most of the epithelial cells that viruses are first going to infect in our respiratory, gut, or genital epithelia or mucosal epithelia, these epithelial cells will be terminally differentiated, not dividing. So most viruses are going to infect a cell that is not really actively in the cell cycle. So, you know, host enzymes for DNA replication might be available, but they're not really present initially. So then there's a choice for viruses. Are you going to induce the cell cycle? So if the answer is no, then that means that you've got to have your own enzymes for the DNA replication process. You can use host cell enzymes for mRNA transcription and maturation, but you have to have your own enzymes for DNA replication. So that's true for herpes viruses and other large DNA viruses that infect insects like the baculoviruses. So on the other hand, if you say, yes, let's go for it, I'm going to push the cell into the cell cycle so that it's going to produce deoxynucleotide triphosphates, produce DNA polymerase, and all the accessory proteins and enzymes required for DNA replication. And that means the host enzymes become available for the DNA virus. And you don't need to code in the genome for all this extra stuff required for DNA replication. So small DNA viruses, polyomaviruses, papillomaviruses, and adenoviruses, they all have the requirement really to push the host cell into the cell cycle. Because otherwise, they can't really replicate their DNA because the host cell will not have all the material available. Yeah.
Those three viruses can still replicate if the cell is already in cell cycle, or does it have to be induced by them?
Yeah, so if they are lucky enough to infect a cell that's already dividing, it's easy for them. They don't need to do anything. But in most cases, most of the cells they infect will not be actively dividing. Okay. So that's a kind of general situation. So the things that viruses need to do really depend on this kind of decision process, and the more independent the virus is, then the bigger the genome has got to be. Okay, so we're just going to look at one example of a small DNA virus and one example of a virus with a larger DNA genome. So the example for small DNA viruses are going to be papillomaviruses. And the reason, so these are non-enveloped T=7 icosahedral capsids, and inside you've got a double-stranded circular DNA genome, just 5 kilobases of genetic information in there. Now, the reason this one is often used as an example, it's not just because I work myself on the polyomaviruses, but it's because, this was really the first virus that was studied to try and understand how it replicates. It was a model system for the replication of DNA in eukaryotic cells because it's got such a simple genome. So a lot of what we know about these viruses comes from studying the simian virus SV40. And so, in a lot of textbooks, SV40 is used as the model example. So these viruses seem to, in the human viruses anyway, they infect us during childhood, don't cause a symptomatic infection. You probably, all of us, or maybe 90% seropositive for the BK polyomavirus, but you won't know it. So they give a lytic primary infection, but they can remain latent or in a kind of inactive state in the kidney. So we're all probably persistently infected by BK and polyomavirus and maybe JC. So, okay, so they either persist at a low level in infected cells, or they go to a lytic cycle, but they are generally not oncogenic. They are not directly associated with human cancers, these viruses. Now, the example for large DNA viruses are kind of a bit of a funny. I'm finding it a bit difficult to say this because the actual situation is a little bit more complicated than that. But, well, they can occasionally cause cancer by accident. Do you want to hear about this, even though it's off the program? Okay, so it's one of the things we're interested in, okay, is that So this, the BK polyomavirus, it's latent in the kidney. It can be a problem in people who get a kidney transplant. And so among people who get a kidney transplant, some of them will have reactivation of the BK polyomavirus. It comes from the transplanted kidney and the virus can reactivate. So one of the things that can happen is this can be a problem for the kidney graft, can be destroyed. So this is called polyomavirus associated nephritis or nephropathy. But even if this can be managed by the medical team and you know the kidney stays in place, one of the things that has been noticed is that the patients who've had a kidney graft and they have BK virus nephropathy, they've got a higher incidence of uroepithelial cancer. So this can be in the bladder or the urothelium further down in the, you know, like in the, you know, the urological tract. So, and when you look, some of these cancers, they have got BK virus DNA associated with them. Okay, but this is rare. This is a rare occurrence. Like out of all the many millions of people who are carrying BK polyomavirus DNA who've been infected, I don't know how many cases there will be in France. Maybe a few hundred per year or something. Okay, so it's a rare occurrence. So generally not oncogenic, generally is true, but there can be cancer in some rare cases. And what is happening here is, well, we don't really know, but we suspect what is happening is that part of the genome is deleted and maybe part of it can be integrated into the host chromosome. So we'll maybe talk about this a little bit later. So there are examples for the large DNA viruses are herpes viruses. So these are envelope viruses with an icosahedral capsid. On transmission electron microscopy, they kind of look like this. So this is probably an artifact because of the sample preparation. On cryo-electron microscopy, they look like this. So probably the real particles look like this, not really like that. So the much bigger genomes, 120 to 235 kilobases in length. And the way the genomes are organized is very different between the alpha, the beta, and the gamma herpes virinae. So these are the subfamilies. Okay, big genome, it's going to be complicated. One of the, normally I wouldn't present this, but it's going to be important, I guess. So for alpha and beta herpes viruses. Bonjour. Ah oui, volontiers.
Merci. Bonjour à tous. Je me présente, je m'appelle Tristan, je suis militant à l'Union Pirate au syndicat étudiant. Et donc, vous avez sûrement entendu parler, mais en ce moment, c'est les élections étudiantes. Ces élections, elles sont très importantes parce que ça nous permet d'élire nos représentants au conseil d'administration, mais également au conseil académique. Le conseil d'administration, il définit toute la politique de l'université de A à Z, son budget et toutes les positions politiques que va prendre l'université. Le conseil académique, c'est plus sur la formation, ça va être le fait que les licences soient, qu'il y ait une compensation ou non, ou qu'il y ait, par exemple, la compensation, le nombre de places en licence, en master, etc. Donc, c'est très important d'avoir des élus qui luttent pour avoir, par exemple, des places en master. Je ne sais pas si vous avez déjà en master, mais voilà. Et donc, ces élections sont très importantes. Nous, l'Union Pirate, on se mobilise aussi parce que les étudiants sont attaqués par des politiques qui nous précarisent, avec, par exemple, un étudiant sur 3 qui vit sous le seuil de pauvreté, un étudiant sur 2 qui est obligé de travailler à côté de ses études. Pourtant, c'est le premier facteur d'échec. Et donc, nous, pour tout ça, on se mobilisera pour le gel des frais d'inscription, pour la suppression de la CEVEC et globalement pour une université plus accessible. Une autre raison de se mobiliser, c'est le fait qu'il y ait 2 listes d'extrême droite qui se présentent au conseil d'administration, l'UNI et la cocarde. Ces gens-là, ils font des saluts nazis à l'université, ils agressent les militants de gauche, et donc c'est très important que ces gens-là n'aient pas d'élus, et pour qu'ils n'aient pas d'élus, il faut qu'il y ait un taux de participation le plus fort possible. Voilà, pour voter, c'est très simple. On vous distribuera sûrement des tracts avec un QR code, mais sinon vous avez reçu un mail ce matin sur les gavotes, enfin, 2 les gavotes. Et sur ce mail, vous avez juste besoin pour voter de votre INE. C'est sur votre carte étudiante, c'est pas votre numéro étudiant, mais c'est aussi sur votre carte étudiante. Voilà, c'est très facile. Merci de votre attention. Bonne journée.
Merci. Ah, n'oubliez pas d'aller voter dans les élections locales et nationales, pas que des élections étudiantes. Les jeunes adultes, c'est le secteur démographique qui est probablement le plus faible taux de participation aux élections. Et bon, voilà. Bon. Allez, bon, on va revenir en virus. Donc, l'organisation des génomes chez les herpesvirus. Pour les alpha et les bêta, on a deux séquences ou deux régions de séquences qui sont uniques, c'est ça qui est U, et unique longue. Ah, well, I'm talking in French. That's over. It just happened and I didn't notice. Sorry. So unique, okay, so you've got unique DNA sequences. So the unique sequence long and unique short. And you've got repeated sequences internally and terminally at the genome. So inside the particle, you have a linear DNA genome. So for alpha and beta herpes viruses, you've got terminal repeats at the end of the long unique sequence or the short, and the internal repeats short or long. That's true for the alpha and the betas. For the gammas, 1 second, please. You've just got terminal repeats at the end. Yeah.
Is it the same sequence that is repeated between the alpha and beta?
No, they're going to be different between different species. Okay, so this unique region, this is where the genes are that code for the different virus proteins. So in alpha and beta herpes viruses, all of the genes, they're called UL something or US something. And it's because they're situated either on this part or that part of the genome. Okay. So for examples of alpha herpes viruses, so this is probably not the current official nomenclature now, but herpes simplex virus 1, which causes cold sores around your mouth, is or used to be called HHV1. An example for a beta herpes virus is the cytomegalovirus, called HHV5, and a gamma herpes virus would be, for example, Epstein-Barr virus. So all of these viruses, they cause an acute primary infection. You'll get, if you're infected, you'll get symptoms during the primary infection. The symptoms disappear, but then the virus persists in a latent form. Okay, so herpes viruses, their big specialty is persisting in a latent form and then reactivating. So if you've had a HSV1 infection, the initial site of infection will be around here, around your mouth, but the virus will remain latent in neurons that connect with this region. So it will be the facial nerve ganglion or the trigeminal. And then during reactivation, you get cold sores, you get lesions reappearing at the sites where these nerves make contacts. So beta herpes viruses, another example would be, yeah, so all of these herpes viruses, they stay latent and they can reactivate at a later date. Now, gamma herpes viruses, they are an important cause of cancer. So Epstein-Barr virus was first identified as a, I'm thinking about the disease, it's not Hodgkin, it's the other one. Okay, so it infects B lymphocytes and it becomes latent in B cells, and it's a cofactor, an important cofactor in the development of several B cell malignancies. Okay, so it's an important contributory cause of different types of lymphoma and leukemia. Okay, so that's a kind of background. So what is a latent cycle? A latent replication cycle is what you get, for example, with bacteriophage lambda. So after the initial entry, attachment and entry phases, the genome will be released into the nucleus in this case. But instead of having the full expression of virus genomes, you have an expression of a limited set of genes called latency transcripts, just enough to maintain the genome at a low copy number in the infected cell. So there's, the cell doesn't really show any differences in terms of its function or its longevity. And just carries on there, but at some point, the latent state has got to be reversed or by an external stimulus, which is going to reactivate expression of a large panel of virus genes that lead to active, intense genome replication, and then assembly, and then lytic release of particles. So that's just the kind of like overview of latency, but I'm not really going to talk about, except. Yes, you can be sick after a primary infection, because for another example would be varicella zoster virus. So if you get a zona when you are over 50 or over 60 years old, this is the same virus reactivating that infected you when you were a child, when you were in the primary school. So it's been latent for several decades. And what is happening here is that these kind of reactivation episodes are probably occurring throughout your lifetime, but your immune system is holding them in check. So you'll have cytotoxic T cells that are available, that are able to recognize when latently infected cells suddenly start expressing virus proteins, and they'll come in and kill them, okay? So you probably have this like reactivation, but it's held in check. Then as you get older, your immune system becomes rather senescent, just like everything else, right? And then the virus can, start to, run free. So this is why, older people should get the vaccine against varicella zoster virus, because otherwise you get the reactivation and it can cause symptoms and active disease. Okay, so this reactivation isn't always harmless. One of the reasons, okay, once again getting a little bit off topic here, is one of the reasons that the Epstein-Barr virus, EBV, is associated with cancers is because some of the latency transcripts, they're involved in getting the host cell to survive longer and not be killed. So the virus is kind of like, supporting or its own maintenance, but the upshot of that is, the consequence is that the latently infected cell will not actually be killed when it's supposed to. Okay, so if it has other kind of cancer-inducing events, then the fact that the latent EBV infection is keeping it alive is going to increase the risk. It becomes a full cancer. Okay, so getting back on track here. So thinking about the processes in the lytic infection, okay? DNA viruses got to replicate their genome and they've got to express their genome, okay? So thinking about one example, herpes simplex virus 1 and polyoma viruses. So in order to replicate double-stranded DNA, the virus has got to firstly initiate DNA replication at its own origin of replication. So they need an origin binding protein, an OBP. So HSV has got one, and the polyoma viruses have also got one. It's called the large T antigen. Once the OBP is bound at the origin of replication, it's got to pull apart the two strands of DNA with a helicase activity. So HSV1 does this, and polyomavirus large T antigen can also do this. Once the DNA strands have been separated, then you've got to have synthesis of the primer, DNA synthesis, something that allows the DNA to move along, the polymerase to move along the double-stranded DNA and a whole bunch of other things. And you need enzymes to produce the deoxynucleotide triphosphate. So herpes simplex virus HSV1 has genes that code for all these different processes. So that's why it's independent. Okay, it can make, it can replicate its own DNA. Polyomaviruses, they don't do any of this. They rely on the host genes for all of these functions. So let's have a look at what the large T antigen is doing, because as we said, if the host cell is not in the cell cycle, then the virus has got to induce it so that the infected cell can express all of these enzymes and accessory proteins that are required for DNA replication. And this is another function of the large T antigen, which will bind P105RB protein and the P53 protein. So this will have two effects. One of them to induce the cell to go into S phase, replicating DNA, and it will also inhibit the apoptosis of the infected cell by inactivating P53. Now in permissive cells, where the virus is actively replicating, that's no problem. In the end, the infection will lead to the death of the infected cell. But in non-permissive cells, you can get a dangerous situation where you have an abortive infection, but the cell cycle is still being deregulated by the large T antigen. So this can be the beginning of a cancerous transformation of the infected cell. And that's what we think is happening in these rare cases when you go BK polyomavirus inside the tumor cells, that somehow we've got persistent expression of the large T antigen, but without the rest of the replicated, without the rest of the lytic cycle. So other small DNA viruses have got proteins with similar functions. So adenoviruses, they've got two early proteins. One of them binds retinoblastoma P105RB. One of them binds P53. And in papillomaviruses, similar small double-stranded DNA viruses, two proteins will bind to RB and P53. So E7 and E6. And as you know, human papillomaviruses, mostly HPV16, are the causal agents of cervical cancer. And what's happening is this process. Some of the infected cells get stuck with a kind of defective replication cycle, and this leads to the development of cancer by the E6 and the E7 oncogenes, viral oncogenes. Okay, so large T antigen for polyomaviruses is essential for inducing the cell cycle, and it's also the origin binding protein of the virus. So two hexamers of large T antigen will bind at the origin of replication in the viral chromosome, viral genome, and then it will start to pull apart the two strands with the helicase activity. Once that occurs, then it's cellular proteins that will start to bind to the regions of single-stranded DNA. So RPA, replication protein A, and that's what leads to recruitment of the primase and the DNA polymerase complex. So polyomaviruses, they just start the whole thing off, okay? And then it's the cellular enzymes doing the rest of the work. Yeah, okay, I'll just take a little pause to think about this question here. So herpes viruses, there are different antiviral molecules for different types of herpes viruses, and they are often this type of molecule. So this is acyclovir and this is guanosine. And this is a nucleoside analog. So what do you think is going to happen? Why is this molecule going to work? Okay, who says it's going to inhibit the thymidine kinase? Inhibit the DNA polymerase? Maybe. Inhibit the RNA polymerase? One person. Inhibit the viral primase? Viral RNA polymerase? That's no, because herpes viruses, they use a host cell, RNA polymerase 2. Viral primase could be, but in fact they're DNA polymerase inhibitors. So what's going to happen is that firstly the virus thymidine kinase will phosphorylate here, and then the DNA polymerase can incorporate this into DNA in this place of guanosine. Okay, so this is so you have to imagine it's deoxyguanosine would be here. But what's missing here is you don't have any hydroxyl group of the 3' carbon. So if you incorporate this into a DNA strand, there's nowhere for the next nucleotide to get added. So this is a strand terminator. So nobody's ever tried to develop similar drugs against human papillomaviruses. Any idea why? What's your guess?
Because the virus mutate a lot.
Good guess, but no. Yeah. Herpes viruses have their own virus DNA polymerase, so that's the target for the drug development. Papillomaviruses, small DNA virus, does it have this? Yeah, that's right. They use this host cell DNA polymerase. So they don't have the relevant target for this kind of drug. And that's one of the reasons why it's difficult to get antivirals against papillomaviruses or polyomaviruses. There's no obvious drug target to try and develop drugs for. Okay, gene expression in DNA viruses. I think it'll be okay. So generally, there's a
I just had a question for the drug.
Yeah.
I don't understand how it only focuses the virus and not the information of the cell itself.
Okay, so the question is, how come this acyclovir can target the virus DNA polymerase and not the host cell polymerase? And I guess the answer is, a priori, before you start your drug development pipeline, you don't know, okay? So you probably start with a whole bunch of different candidates and test them. And you're looking for something that will inhibit the virus replication, but won't have any off-target effects on host cell DNA polymerase. And if you've got a different enzyme, then you might get one that has this property. Okay, so you don't know before you start, but you start with the idea there might be some drug that has the capacity to be specific for the virus polymerase and will not inhibit host cell DNA. And of course, it will depend on, concentration and this kind of thing. Okay, so gene expression. So DNA viruses, there's a big distinction between early genes and late genes. So the early genes are expressed before DNA replication, and their functions are to modify the biology of the host cell, like inhibit apoptosis, induce the cell cycle. They can also control the expression of other viral genes and, importantly, initiate DNA replication. The late genes are the genes that are expressed after DNA replication, and these code, for example, for structural proteins and the proteins involved in the assembly and the liberation of infectious particles. So for polyomaviruses, how do they manage this? Okay, so they've got a very, very simple genome, and I'm showing the BK polyomavirus genome, SV40, it's exactly the same thing. So they just have one promoter region. The origin of the replication is here, and it's also the promoter region to express the viral messenger RNAs. And you've got a small genome, so you've only got space for one promoter. So the question is, how does the virus express different messenger RNAs just from one single promoter? Well, it takes advantage of the fact that if transcription factors bind here, then the transcription can either start going this way or it can start going the other way. Okay? And what happens, well, the classical model for polyomavirus gene expression is that initially, you get early transcription in this direction. So you get expression of the T antigens, large T antigen and small T antigen, its little brother. And these are the ones, the early proteins, that will lead to the genome replication. And after DNA replication, you get transcription working in the other direction. And that's what will allow you to express VP1, VP2, VP3, the structural proteins, the capsid proteins, and the rather mysterious agno protein. So small genome, one promoter is used for both the early and the late genes, and alternative splicing is used to give multiple messenger RNAs from the same primary transcript. So you've got alternative splicing here, which can lead to the expression of small, large, and something called middle T antigen. So if you've got unspliced RNA, you get to a stop codon here. But if you splice out this intron, then you don't have the stop codon, you get the rest of the protein going all the way down to here. Okay? Similarly, with the late genes, you've got alternative splicing, which will lead you to express VP2, VP3, or VP1. Not the only mechanism, but that's all you need to know. And the virus is in the nucleus, it can do this, right? Got alternative splicing. How about herpes viruses? So for HSV1, one of the important points is these guys have got big genomes. So they have multiple different promoters that will be active at different steps during the lytic cycle. So initially, it's just the immediate early promoters that will be active. And these will express what are called the alpha transcripts. And these alpha transcripts code for transcription factors. And these transcription factors will initiate the transcription of the next wave of virus genes, which are the beta transcripts, the early genes. So the alpha-4 protein is a transcription factor which will bind to the beta promoters and allow their transcription. The early genes, the beta genes, are all the proteins, or code for all the proteins required for DNA replication. So UL9, UL5, UL8, all these ones that were in the list before. And the DNA replication is what allows the activation of the late genes. And these will be coding for the capsid, the envelope, and the tegument proteins. So what starts the whole thing off? Okay, you've got a cascade, you've got these immediate early genes that allows the beta genes to be expressed, then you get DNA replication that allows the gamma. So what starts it all off? Well, what starts it all off is this tegument protein called alpha transcription initiation factor. And because it's in the tegument, that means that it gets transported, it gets internalized into the cell directly after the membrane fusion step. So it's transferred firstly into the cytoplasm and then gets imported into the nucleus where it can kick off the first round of virus gene expression. So it's not dependent on a host transcription factor, mostly. Okay, so that's two examples of DNA viruses. So now we're going to think about RNA viruses. And I'll give two examples of plus strand RNA viruses, one example of a negative strand RNA virus. Okay, so for plus strand RNA viruses, their basic idea is that they're going to pretend to be a messenger RNA. And so if that's going to work, then the host ribosome has got to be able to recognize the virus genome and translate it into proteins. Among these virus proteins, then one of them has got to be an RNA-dependent RNA polymerase, which is essential for the virus to replicate its genome first into a complementary strand of RNA, which can then be used as a template to regenerate new copies of the genome. So host cell cytoplasm does not contain any enzyme with this activity, and the genome does not code for any equivalent to this either. So every RNA virus has got to code for its own RDRP. Absolutely essential. Okay, so two kind of problems for plus-strand RNA viruses. Firstly, you've got to get the host ribosomes to recognize your genome and translate it. And secondly, one of the problems is you've got multiple virus proteins that you need to express and with different functions, but you've only got one genome plus-strand RNA molecule. How do you get several different proteins from a single RNA molecule? That's the problem. So two different examples, picornaviruses. So I got identified in inverted commas here. So it was the first human disease, or one of, okay, it was it was identified as a viral disease in 1908. Okay, the virus was not purified and studied at that time. Okay, the infection was shown to be caused by a filtrable agent early in the 20th century. So the poliovirus is a member of the Picornaviridae family. So all of these picornaviruses are icosahedral non-enveloped viruses with 7 to 8 kilobase long plus-strand RNA genome. So in picornaviruses that infect humans, there are 3 genera. So enterovirus, cardiovirus, and hepatovirus. Hepatovirus is represented by hepatitis A. And the enterovirus genus is a very, very diverse group of viruses that includes rhinoviruses that cause colds and a bunch of enteric picornaviruses that infect the gut. And amongst those that infect the gut, there is the poliovirus. So poliovirus is unusual because it is also neurotropic and can cause acute flaccid paralysis and, in the worst cases, death. And if you remember from attachment and entry, it's able to do this because its entry receptor is expressed on neurons as well as enterocytes. And we think poliovirus has been endemic in the human population perhaps for thousands of years because we have this stela with an inscription of an Egyptian priest here, and he's walking with a stick and his leg looks like it's a bit strange, it's quite thin and it's held in an unusual way. So this looks like it might be the sequelae or the long-term effects of a polio infection as a child. It's like permanent handicap. So has been endemic in human populations for a long time, almost eradicated. So the other one we're going to look at is flaviviruses, and perhaps in particular dengue virus. So flavivirus are enveloped plus-strand RNA viruses, slightly longer genome. Most of them are in the orthoflavivirus genus. So these are, for example, yellow fever virus or dengue virus, all of which are transmitted by insect vectors. So which will be the Aedes mosquito, right? I believe. So the other important flavivirus as a human pathogen is hepatitis C virus. which is not insect transmitted. So these orthoflavivirus infections, they're kind of interesting because you have an animal reservoir and the virus can spill over into humans through the vector, okay? And so the problem for control is, even if you eliminate the transmission in the human population, you're always going to have reintroductions from the animal reservoir. So it's not an endemic virus in the way that polio is. So we can never really eliminate these guys, but there can be effective human-to-human transmission. Okay, so you've got what's called a sylvatic cycle. There's the animal reservoir and occasional transmission, but you can get sustained chains of transmission human-to-human through the vector, because especially with yellow fever. Hepatitis C, well, that's kind of different case. Most of the transmission is bloodborne. So these orthoflaviviruses, you just have an acute infection, and either you recover or you can die, okay? So those are two options here available. And if you recover, you have long-term protection, and that's why vaccination can be effective. Hepatitis C, maybe 20% of people who get infected will eliminate the virus, 80% will go on to have a chronic infection, and which could lead to fibrosis and liver cancer. So it's another cancer-inducing virus. Okay, so how to get the host ribosome to translate the genes encoded in the virus RNA. So if you look at picornavirus genomes, structurally they are unusual because the 5' extremity of the virus RNA does not have a 5' cap structure like a messenger RNA. It's attached to a virus protein called VPG. And if you look in the genome where the proteins are coded, there's a 5' untranslated region which contains start codons. So translation doesn't start from the 5' end of the genome here. It actually starts later on, downstream of this region, which is called the internal ribosome entry site, the IRES. So ribosomes can bind to the IRES and start translation from the first AUG, which is downstream of this IRES site. So why does this work? Well, the IRES is characterized by a lot of secondary structure. So there's a kind of three-dimensional structure of RNA, and the ribosome binds to that. So that's kind of, well, useful. One of the consequences of this for picornaviruses is that they can shut down host cell protein translation by cleaving and inactivating the eukaryotic initiation factor 4G, which is the initiation factor that binds to the 5' cap on messenger RNAs and starts to recruit the rest of the ribosomal subunits. So the virus can degrade this essential host cell protein. So the host cell can't express any proteins anymore. But the virus, it doesn't care because the ribosomes are going to come in and have cap-independent translation. So that's very useful for the virus to completely take control of the host cell translation, protein translation machinery. So that's picornaviruses. How about flaviviruses? Well, it depends on the virus. So for hepatitis C virus, which is illustrated here, you have a similar kind of thing. The translation is also iris dependent, although the secondary structure is not related at all to what you find in picornaviruses. Whereas in dengue virus, you have a mixture. It can be cap dependent or cap-independent translation. Okay, so that's how translation occurs. The next problem for the virus is how to get multiple different proteins from the same RNA molecule. And picorna and flaviviruses both work in the same way. They express a big polyprotein that covers practically all the genome. And this polyprotein is then going to be cut into individual proteins by the viral proteases. So in picorna viruses, like poliovirus, it's the 3C and the 2A proteases. Another thing that I think is worth noting is the way the genes are organized on the virus genome. So you've got, firstly, the structural proteins here in the 5' region. Okay, so VP1, VP3, VP2, these are the capsid proteins. Then at the other end, you've got the RNA polymerase, the protease, well, the main one, and the VPG protein. VPG and the RNA polymerase essential for RNA replication. And then in the middle, you have these accessory proteins doing something else. But the two really important functions, capsid and RNA polymerase, they're at opposite ends of the genome. Okay. I don't really know why that is, but it's true for all picorna viruses. How about flaviviruses? Well, as I said, it's a similar kind of thing. You've got the polyprotein, which is then going to be cleaved mostly by the virus NS3 protease, but with some contribution to signal peptidase and the Golgi peptidases for the envelope glycoproteins and NS1, which is also going to be in the kind of lumen of the endoplasmic reticulum and the Golgi. So similar kind of thing, polyprotein and then cleavage. And once again, you've got similar kind of organization because you've got the capsid genes, the structural proteins in the 5' end of the genome. NS5, NS5A, that's the RNA polymerase. So RNA replication and then the protease, they're at the kind of 3' end of the genome. 5' end structural proteins, and then somewhere in the middle, accessory other proteins doing other jobs. Okay, it's midday. Yeah, we can do negative strand RNA viruses, might leave retroviruses till next time. Okay, yeah, I think we'll do that. We'll just do negative strand RNA viruses. Okay, so negative strand RNA viruses. So their genome, unless my animation is no good, doesn't code directly for proteins. So what these viruses have got to do is they've got to replicate their genome via a full-length transcript. So this is an RNA copy of the genome. And they also have to express messenger RNAs. And these messenger RNAs, okay, will have 5' cap and a poly A tail, which allows them to express proteins. That's not the case for the genomic RNA. So the main issue for negative strand RNA viruses is that what enters the infected cell initially is this, the negative strand RNA genome. And for anything to happen, the virus has got to be able to transcribe the genomic RNA either into a messenger or into another copy of its genome. And that requires an RNA-dependent RNA polymerase, an RDRP, to be present in the virus because the cell doesn't have any RDRP hanging around there. So all negative strand RNA viruses have got to have an RNA polymerase that comes in together into the infected cell. That's not the case for plus strand RNA viruses. Okay, so for negative strand RNA viruses, what are the problems they have, how to make messenger RNAs that the host cell ribosomes can use, and how do you get multiple mRNAs from the same genome? Because that's not inherently obvious, because it's an RNA molecule. You don't have a promoter and you start your transcription from the promoter region. You always start from the beginning. Okay, it's not obvious how to get multiple mRNAs. So the example that I prepared is the orthomyxoviruses, which include influenza viruses, cause seasonal respiratory disease. Influenza A, B, and C infect humans. Tagotoviruses are related, but they are tick-borne and don't cause respiratory illness. Out of the A, B, and C influenza viruses, influenza C is asymptomatic. Influenza B, considered to be infecting only humans. And influenza A viruses are the ones that can infect humans, animals, and birds. So the influenza A viruses are the ones that have a pandemic risk. So these viruses are enveloped with a helical nucleocapsid, and inside each virus particle you don't have a single molecule of genomic RNA. You've got 8 segments which are ordered, which are numbered in the order of decreasing size. So segment 1, that's the biggest RNA molecule. Segment 2, the next biggest, down to segment 8, which is the shortest. And generally speaking, each genome segment will code for 1 mRNA molecule and give you 1 protein. The exceptions are the small ones, 7 and 8, which can produce 2 proteins by alternative splicing. So what are the genes coded here? So the biggest gene segments, 1, 2, and 3, code for the RNA polymerase subunits. So P is polymerase, PA is polymerase acidic protein, PB1, PB2, polymerase basic 1, polymerase basic 2. So basic, they've got net positive charge, they can bind to nucleic acids easily. Then you've got structural proteins, the nuclear protein, which associates with the RNA to give the nucleocapsid as kind of like baton or stick-shaped helicoidal capsids. HA, the hemagglutinin, the binding protein, neuraminidase, glycoprotein for release of the particle. M1 and M2, also structural proteins, the 2 matrix proteins in the virus, and NS1, NS2, non-structural. Okay, all of these other proteins, you find them in the virus particle. Nucleocapsid, and then the polymerase subunits are associated with each genome segment. So each genome segment, you've got the nuclear protein, and you've got 1 copy of PA, 1 copy of PB1, 1 copy of PB2. In the envelope, you've got these guys and the matrix proteins. So one of the things that influenza orthomyxoviruses, they've kind of come up with what I think is a bit of they've applied lateral thinking to this problem. How do we express several genes from several different proteins from our negative strand mRNA genome? Their solution is, hey, we just cut up our genome into different bits, and then we get 1 protein per segment. Easy. They've solved the problem. Now, how about the 5' cap and the poly A? How to get messenger RNAs? Well, orthomyxoviruses, they are exceptional amongst negative strand RNA viruses because, well, for all RNA viruses, because they replicate in the nucleus. So this is what gives the virus access to the splicing machinery of the host cell. That's why you can get 2 alternatively spliced messenger RNAs from these segments. But it also gives the virus a mechanism to get caps on the 5' extremity of their messenger RNAs. And they do this by a mechanism which is called cap snatching. So the functions of the polymerase subunits PA and PB2 are to cleave off the 5' few nucleotides of a messenger RNA in the nucleus and then transfer it on to PB1. So this is a kind of diagram here. So PA and PB2, they've got this 5' cap from a cellular messenger RNA, and it's being passed, and this will be used as the primer to make a copy of the genomic RNA here. A messenger RNA will be copied off the virus RNA, which starts down here. And then it will be extruded, well, the template comes out this way, and the new messenger RNA will come out this way, and you get dNTPs added at the base. So that's how orthomyxoviridae produce messenger RNAs. So I'm not going to do retroviruses because we maybe don't have time. So what I think I will do is, yeah, and the next lecture is less complicated anyway, so we've got a little bit of time here. So also talk about other negative strand mRNA viruses. So okay, so orthomyxoviruses, they found a kind of sneaky way to solve these two problems. How to get several proteins off one RNA molecule and how to get messenger RNA. So there are a lot of negative strand RNA viruses that just have got one big, long negative strand genome. So for example, Ebola virus, measles, rabies virus. Okay, and they generally are quite nasty, these infections. Okay, I don't know why, but these mononegavirals, they are all pretty nasty. How do these guys work? They replicate in the cytoplasm. They don't replicate in the nucleus. And now I'm going to have this negative strand genome represented 3' to 5' here. So they have their own polymerase, which is called, rather confusingly, the L protein for large protein associated with the phosphoprotein, which is P. So what they do is they start binding here, and they start producing a messenger RNA. So this will be 5' end here. And they'll start transcribing to produce this messenger RNA. Actually, I should probably draw this the other way. Now, how about the cap mechanism? This large protein, the large polymerase complex, has got the ability to add a 5' cap. So it starts producing the messenger RNA, then it adds on the required modified nucleotides. So it has a cap synthesis activity. Okay. Then what happens? Then you come to a region which has got several uridines. I think it's like 7 or something. 1, 2, 3, 4, 5, 6, 7. And once it gets to this polyuridine tract, 7 of them, what will happen is it's going to get here, then it will slip back, then it will get here, then it will slip back, then it gets here, then it will slip back. And so that will give you a poly A tail. So this happens for a while, and then what happens is the polymerase will detach. It detaches. And then one or two things can happen. One is that either it will bind back and it will start to produce another messenger RNA from here, or it will go all the way back to the start. Okay? So it's got this mechanism where the polymerase will transcribe one part, add a poly A tail, then be released, then restart again. And so you get like several different messenger RNAs coming from the same template, negative strand, complementary strand RNA molecule. So if you had to guess, this transcript or this one, which are you going to get the most numbers of copies of? Yeah, so you get a lot of copies of this guy and progressively fewer copies of the others. Okay, so what's this gene going to code for in the mononegaviral genome? The one you need the most copies of? Capsid, the nucleocapsid protein. So this gene is going to code for the nucleocapsid. And this one, what do you need the fewest number of copies of? Something that is an enzyme, it's got an enzymatic activity, can make loads and loads of copies of something. Yeah, RNA polymerase. So this will be the L gene, okay? So mononegavirals like these ones, they have a different strategy to replicate, okay? So that's for the mRNA production. One of the questions is, how do you make a genome? Because in order to get the genome, you've got to have a full-length transcript, and you don't want to have all this poly A screwing around with you. So what happens here is that, yeah, once you get enough nucleocapsid which is accumulated, then this binds to the mRNA as it's being produced, and it will suppress this mechanism for adding the poly A, and it will suppress the detachment of the polymerase. So once you've got enough nucleocapsid protein, then your polymerase goes all the way to the end and produces a copy of the genome. Yeah. I think because as soon as the template is like being produced, the nucleocapsid will bind, and it will prevent it from like slipping back or making base pairing with the U region here. The first A's that come across. Okay, I don't know exactly. I'm kind of making this up, all right? But what I imagine is, as soon as these A's are synthesized, they'll be bound with the nucleocapsid, so they can't like participate in base pairing, which is going to cause the slipping of the polymerase complex. Yes, I get that. Yeah, all of them are required, but in quantity, you need more copies of this protein than you need copies of the polymerase. So you need like maybe several thousand copies of the nucleocapsid to get around one genome. But you only need like one copy of the polymerase per copy of the genome that you're making. So it makes sense to have the genes organized this way on the virus genome. And that's also highly conserved. All the mononegavirals have got their genomes organized this way. And it's not an accident, it's like optimized. Okay, so I guess that is it. And so 2 p.m. we meet. Where in what room is it? 209. 209. So I'm going to go home, get my power cable, and then come back. And so see you all after lunch, I guess.